I'm Yosef, and my project is the neurotrophic effect on embryonic stem cell growth. Basically, neurotrophins are really just this growth factor that helps cells in differentiation and survival. They're first created as proneurotrophins, but then they end up dimerized together to create the neurotrophin. There are 4 different types of neurotrophins, as you can see, and they bind to the P75 receptors and TRK receptors, but I'll be focusing on TRK receptors. As you can see, there's always a complicated signal transduction pathway no matter what you do, but to keep things simple, basically it either grows or dies. Alright, so we know that stem cell or the neurotrophins aid in survival of the cell, except do they do this by mitosis or by terminating apoptosis? And so in order to do this, I grew everything on the same conditions. And you can see I grew my embryonic stem cells on embryonic feeder cells. And here's a picture of the stems of the MES cells right here. And these stringy looking cells are the feeders that they're growing on. And so I had a condition where they were growing with neurotrophin. You see the effects of the neurotrophins. Cells where it didn't have anything with them, it was just, I guess, a wild type. And this should say neurotrophin inhibited by antibodies, so I apologize for that. And this control antibody condition where I just put in general antibodies to see if it's the antibodies in general that are affecting the mitosis of apoptosis. And I have negative control right here. Alright, and to measure mitosis, I did immunohistochemistry through the phosphates in the histones, which are created while the cells undergo mitosis. Just keep it simple, I'll leave it as that and because of time. And so what I did is I put in primary antibodies which attach to the phosphate groups within those histones inside the nucleus, and then I added secondary tagged antibodies which are fluorescent. Here are the pictures really. Right here, this is a condition where the neurotrophins are inhibited by the antibodies. The control antibody, my neurotrophins, my untreated cells, and my negative control. It might be a little bit hard to see, but where you see the red is where the mitosis is occurring, and the secondary antibody fluorescent tag is seen, and the blue is the stain on the nucleus within the cell. As you can see from my data, the neurotrophins indeed help out with mitosis. Alright, and in order to measure apoptosis, I did a different procedure, which is called tunnel assay. And basically what happens is when cells undergoing apoptosis, there's this enzyme that actually breaks apart the DNA, and so you have little fragments coming off. And then I introduce another enzyme that puts tagged fragments of DNA back in there, and so I'm able to go back and, under the microscope and look at these fluorescent tags I put back into the DNA, and this is what you see. It's almost the same thing as the PHH3, but in this case, the fluorescent tagging is green. I know the pictures might be a little bit hard to see, but as you can see right there, there's green right there, and there's really green everywhere. And once I put all the data together, in fact, the cells that had neurotrophins indeed have the least amount of apoptosis occurring in them, which means neurotrophins do, in fact, help terminate apoptosis. Okay, and so my conclusion is basically right there. Neurotrophins aid in mitosis, and neurotrophins aid in termination of apoptosis. Wait, sorry, they aid in mitosis and help terminate the apoptosis. I think I'm going to use some words. Anyways, now connecting back to cancer. TRK receptors are really a huge thing to look at in cancer because, well, the TRK receptors were really first discovered as a proto-oncogene. And furthermore, since I'm looking at the survival of cells, that's really a huge, that is cancer right there. It's uncontrolled growth in cells. And so it was meant to find a way to properly inhibit or actually work with these attractor receptors so we can control the growth of these cells. We'll actually be doing a huge thing in the further research in cancer. And what to do now? Now we got to figure out exactly how to find the proper sequence or the proper signal pathway to either inhibit or induce in order to prevent the cancer and in order to get the results we want. And now finally the acknowledgements. Thank you to Dr. Leslie Locke for all your help, and of course my mentor, Dr. Sarah Winokur, and my graduate student mentor, Noel Thompson. She couldn't be here today, and of course, thank you to the rest of the Locke Lab and my school staff from the high school I went to, but unfortunately they're not here. And of course, the UCI Cancer Research Institute, Beckman Coulter Foundation, and the American Cancer Society for such an amazing opportunity. And thank you, General.